Hi everyone, welcome to Manathi's Surprise Weekly Liquid Biopsy, Part 17 Circulating Tumor Cell Journal Club podcast. From now on, I decided to continue the podcast with good impact PubMed index literature for future high indexed science backed products. Today, let's see a recent review published in BBA Reviews of Cancer with the title Circulating Tumor Cells, the Good, the Bad, and the Ugly, published in February 2023. Due to time constraints, I'm reading out the key takeaways. The good, the clinical utility of circulating tumor cells (CTCs). The good includes their diagnostic, prognostic, and therapeutic potential. The bad, the complex biology. The bad, CD45 positive, EpCAM positive CTCs means CD45 white blood cells interfere with CTCs, which is crucial for the purity and isolation of CTC and its spheres for drug development, limiting the clinical translation. The ugly, the microemboli are ugly means dangerous CTCs with EMT and MET phenotype forming homotypic and heterotypic clusters with immune cells and platelets, increasing the metastatic potential. To note, CTCs were even included in the 8th edition of the AGCC TNM staging system for breast cancer. The detection of greater than 5 EpCAM positive and CD45 negative morphologically viable CTCs in 7.5 ml of blood CellSearch system correlates with both reduced progression-free survival and reduced overall survival in patients. The CellSearch system depends on the chemical fixation immunostaining cell surface marker for CTC isolation. The fixation step destroys certain morphology molecules, example studying the secreted protein like SFRP4 and SFRPs attached to the cell. For this reason, scientists have developed a microfluidic system for label-free viable CTC isolation by Vortex Biosciences, like our emerging preclinical STEM CTC technology system, with the advantage of automating and isolating in large culture volumes for human protein medicine therapeutics. This clinical threshold also distinguishes patients with malignant disease, as CTC events are typically rare in benign patients and healthy controls. Label-free enrichment devices may capture a greater diversity of CTCs within patients but may be prone to higher rates of false positive detection and contamination from circulating leukocyte populations. CTCs may also offer a route for preclinical drug screening through ex vivo culture as cell lines or spheroids or expansion in vivo models. Evidence suggests that molecular exchange between interacting platelets and CTCs also promote a more stem-like phenotype in CTCs, further enhancing their metastatic potential and ability to survive circulatory shear forces. Targeting the interaction between platelets and CTCs with therapies such as aspirin may help prevent and treat cancer metastasis. The aspirin drug response on CTC sphere is patented by a Singapore group. CTCs at single cell resolution methodology has been successfully utilized to generate single cell profiles. Such as approaches are laborious, low throughput, and difficult to scale up. Massively paralleled single cell sequencing approaches also allow for greater standardization of CTC sequencing and validation of CTC in future studies, as we intended to develop radiotherapy CTC kinetics therapies. Single cell analysis of increased number of CTCs will likely also allow for more comprehensive profiling of the phenotypic diversity of CTCs within individual patients and intra-patient heterogeneity. The take-home message: traditional surface marker based detection system such as the FDA-approved CellSearch device have shown clinical utility as a diagnostic prognostic test for metastatic prostate, breast and colorectal cancers. The Genostics Maintrac system successfully tests drug response on patient CTC sphere samples in the Australian market. Still, the Medicare system does not cover the testing expenses. India requires such technology to treat their patients with quality of life. Thank you. See you in the next Journal Club podcast.